All right, very quickly, we're going to go through the procedure for the P-Glow lab. All right, the first thing you will get as you gather your materials together is a plate, a petri dish that has gelatin called agar on the bottom. It's a nutrient agar, and on this plate you will see a cream-colored some cream-colored streaks and also some individual spots. All of these streaks here and here and here, this stuff in the middle is just condensation, water droplets on the plastic plate itself. But these spots are individual colonies of bacteria that are growing on the nutrient gelatin or agar. This is our bacteria that we'll be trying to transform, okay? Please note, this is a colony. These are not individual bacteria. There are millions of bacteria in each colony. So you will be asked to collect a colony or two of bacteria. You truly do not need to collect more than whatever it is you are asked to collect, because there are millions of bacteria in one of these little spots, okay? Each spot represents a place where one bacterium was about 24 hours before, and this represents how much has grown over about a 24-hour period, okay, from that single parent bacterium. Okay. All right. You will also get some test tubes, 4 of them, and a little floating foam rack. 2 of them, the yellow and the kind of grayish clear tube, will contain substances in them already, liquids. This will be labeled LB, and it will contain something called Luria broth, which is a liquid nutrient. It's the same stuff that's in the gelatin in the bottom of the plate, but this is just in liquid form. It's a nutrient, nothing more. Okay, nutrient. In this tube, however, is that stuff that we talked about previously called CaCl2, calcium chloride. This is the transformation solution. Okay, and so this is what we are going to put the bacteria in, and as we are trying to encourage them to pick up the plasmid. So this will be labeled CaCl2, and it's your transformation solution. You will also have 2 other microtubes. You will need to label one of them as plus and one of them as minus, okay? These are going to be for your 2 different treatments in this experiment, okay? You will be asked to place bacteria in each of these and some different substances in each of these. Let's talk about what you will have, okay, to work with. I do want to point out to you the droppers. You'll be working with these pipettes that you've worked with before, but I do want to bring you to mind what the graduations are on here. 1 ml is the line at the top. We don't really care about those. The next line we care about, actually, let me show you this one first. You see how the pipette goes inward right here? That mark right there is the 100 microliter mark. You need to know that. And then the next mark above it is 250 microliters. So you need to be aware of where to find those marks when you need to. Another piece of material that you will have to work with are bacteria loops. These are little wands that have a very, very tiny loop on the end that is used for collecting colonies of bacteria. Please keep in mind with these and the pipettes this little slogan: when in doubt, throw it out. The materials that we work with need to be clean. They need to be as sterile as possible. Okay, they need to be clean. And so if you're not sure if a loop or a pipette has already been used or if it's touched something like your hand, then just get rid of it. We've got plenty, so keep that in mind. There's the tiny little loop on the end, and you will use these to collect bacteria off of those plates, like what we looked at the beginning of this right here. You will take the top off of your bacterial plate, and you will just lightly scrape with the loop, lightly, a colony or two, however many you're directed in the procedure. You do not gouge into the gelatin that's in the bottom of these plates. You just lightly scrape off a colony or two and put it in to the vessel you're asked to, which is going to be one of these, or actually each of these tubes on this side. Okay, so here's your experimental setup. Initially, you're going to place calcium chloride from here into each of these tubes, and your procedure will tell you how much to add. So you're going to put calcium chloride in there. Then you're going to place bacteria in each of these tubes. So far they're identical, bacteria and transformation solution, calcium chloride in each tube. However, in the plus tube you will also add a helping of P-Glow plasmids. Okay, now let me show you what I mean by that. I will have a vial. You will not have this. I will have this. This contains down in the bottom a little tiny bit of fluid that contains our plasmids that have been engineered for us, that have the P-Glow gene, the gene for GFP, the antibacterial gene, and so on. You will stick a clean loop into this vial. You will twirl it around, and your goal is to get this like you were about to blow a soap bubble. You will cover your loop with the solution from this little vial, and this material will contain thousands of P-Glow plasmids in it. Okay, and then you will take that that loop, and you will stick it down into your plus microtube, and you will twirl it around in there to try to get as much of those plasmids off of that loop as possible into your plus container. Okay, so now what you have are bacteria in transformation solution that aren't getting the plasmid, and you have bacteria that are getting the plasmid. Okay, everything about these two tubes is identical except for who's getting the plasmid and who is not. Obviously, I hope you can recognize one of these as the control treatment and this over here as the experimental treatment. Okay, we would expect nothing unusual to happen to the ones that aren't getting the plasmid, and we would expect these to behave differently. We'll see if they do. Okay, now once you have done that, you will have another foam rack, and you will place your plus microtube and your minus microtube in this rack, and you're going to come back to where you're actually, I'm ahead of myself, sorry. You will place these on ice for 10 minutes. This is the first step of the heat shock. Okay, you're going to get them cold for 10 minutes, making sure that the ends of the tubes are down in your ice bucket that you will have. Once 10 minutes are up, you're going to come back to a water bath, and that's what this is in the back of the room. There's another one. It looks just like this. They will both be the same. They will both be at 42 degrees Celsius. Okay, so 42 degrees Celsius. You will, the lids will be open. You'll see water standing in them, and you will float your little test tubes on their little block, in their little foam block, in the water here. One plus, one minus. Okay, and you will do that for no longer than 50 seconds. Okay, 50 seconds, and then you will have your ice bucket with you, because if you do not have your ice bucket with you with the ice in it, it's going to take you too long to get back to the table. You won't shock them. So what you have to do is to get them out of the ice, put them right in the hot water, then put them right back on the ice. So that's going to provide the heat shock. So you let them sit in the water for 50 seconds, then back onto the ice, and then you'll let them sit on the ice for about 2 minutes, and then they're ready. They're on the lab counter ready to go. Now, in addition, you will have at your disposal 4 dishes that have agar on them. They have various abbreviations on the bottom. LB means Luria broth. It's a nutrient. There's nothing else in this agar but nutrient. This one has nutrient, as you can see, but also AMP, which stands for ampicillin. There is ampicillin in this nutrient as well as just nutrient. This one too, and this one's got ampicillin, nutrient, and another nutrient that is labeled Ara for arabinose, and that is a sugar that some bacteria can readily consume. Okay, we'll talk about why that's there in the next podcast. Okay, but those are your Petri dishes. Your job will be to first label the Petri dishes as follows. Okay, you will be labeling them. I want to make sure I've got it right. One will be labeled as minus P-glow. That's the LB, the plain LB. One of your LB amp plates will also be labeled as minus P-glow, and then one will be labeled plus P-glow, and the arabinose plate will also be labeled plus P-glow. Okay, obviously what this tells you is you're going to put some of the bacteria that didn't get the plasmid on this plate, some of the bacteria that did not get the plasmid on this plate, some that did on this plate, and some that did on this plate. These up top are your control plates. These down here are your experimental plates. Okay, and we're going to compare them to each other. Now, what we might want to talk about is what you might expect. Do you think that normal bacteria can grow on a normal nutrient plate? Okay, probably. So this you might expect some growth on. Can normal bacteria with no resistance gene grow on an ampicillin-laced plate? Think about that. Should bacteria that have a gene for ampicillin resistance, which the P-glow plasmid will provide, be able to grow on an ampicillin laced plate? Should we see growth on this plate with the engineered bacteria? Should we see growth on this plate with the engineered bacteria? Okay? Think about that. All right? Now, in order to find out answers to these questions, we're going to have to let these bacteria grow. Okay, so you will place them. Once you're done, you're going to tape them all together in a little stack and identify them, and you're going to put them in this incubator for overnight. Okay, and that's that. That's the general idea of the lab. You'll get a procedure, follow the procedure in class, and you should be good to go. This lab works really well, and then we'll talk about the results you should see next time. All right, thanks for watching.